Welcome back to another exciting episode of Inside Learning. Today we are going to discuss about transduction in bacteria. Before we can proceed on the types of transduction of bacteria, let's discuss what is the process or what is how we can define the transduction in bacteria. Simply imagine two bacterial cells that are present in the same generation. There are two conditions, one bacterial cell and the other bacterial cell. Suppose they are present in the same generation, so one bacterial cell transfer its own chromosomal material or its own genome to another bacterial cell with the help of virus. This is the condition. The first condition is they must be present in the same generation, and the second condition is the transfer of genome is taking place with the mean of virus. So this type of transformation or this type of transfer, we call it as transduction, and because it is taking place in bacteria, we call it as transduction in bacteria. Now the transduction in bacteria, it may be generalized transduction or it may be specialized transduction. Discuss the whole process so we can easily understand these terms. In the generalized transduction, first of all, suppose there is a bacterial cell. We all know that the bacterial cell is affected by the virus. It is named as bacteriophage. The virus which affects bacteria is named as bacteriophage. In this step, simply I am here representing the genome of bacteriophage. This is the virus that is affecting. In this phase, in this step, the virus is affecting or transferring its genetic material into the bacterial cell, and there is the purple color, the red color representing the genome of bacteriophage, and the purple color is representing the genome or the genetic material of bacterial cell. Here the A plus and B plus means these are the genome or these are the genes that are present in the cell of bacteria. So what happens in the next step when the virus completely injected its genome into the bacterial cell? Ultimately, when the genome is completely transferred, the virus genome is completely transferred, the host DNA is hydrolyzed, the bacterial DNA is hydrolyzed, and it means it breaks down completely into pieces, and the phage DNA and proteins are made. Then ultimately, when the virus DNA completely destroyed the whole chromosomal structure of bacterial DNA, then in the next step, the virus genome is starting to replicate or form the new structure of viruses. How? Here we can see. So this is the turning point. Here you can see that when the new bacteriophage structures are formed, ultimately these are the normal structure. This is bacteriophage, this is bacteriophage, this is bacteriophage. But the genome that is present in this bacteriophage is different. The genome that is present in this bacteriophage is the genome of bacteria. Here I am representing bacterial genome as purple color and phage genome as red color. Occasionally what happens, a bacterial DNA fragment is packed in a phage capsid. So what will happen next? When these bacteriophages are released, all of them are going to affect some other bacteria. This one bacteriophage is going to affect another bacteria, this one is going to affect another bacteria. Then what happens? Bacteriophage is going to affect some other bacteria. What happens? It will transfer its bacterial genome to the bacterial cell, to the new bacterial cell or second host bacterial cell, you can say that. So this is A positive because it is bacterial genome. It is not viral genome that is going to affect the bacteria or starts a lactic cycle or starts breaking down the genome of the host bacterial cell. It will drive the A positive, or it will transfer the bacterial genome of A positive gene into the new bacterial cell, which has which has already A minus and B minus. It means both of these genes are absent in this bacterial cell. But when the virus transferred the A positive into the bacterial cell, ultimately A minus is converted into A positive, and they start A positive start integrated into the part of this gene of bacteria. and A positive is start integrated into the part of the genome of second bacterial cell. So what will happen? A positive, B minus. What is that mean? What does that mean? It means A positive is the genome, is the genetic material, is the gene sequence that is now integrated into the main genome of the second host bacteria, and the B minus belongs to its already or its original chromosome. So this is the new bacterial cell. This is the recombination. This is the new genetic material. This is generalized transduction. We can define it as the transfer and packing of pure bacterial DNA. Transfer and packing, what happens in this step? The packing of genetic material of bacteria, the pure form of genetic material. There is no accumulation of virus DNA. There is only presence of bacterial cell DNA in this capsid. So the pure form, the pure bacterial DNA, when packed and transferred into the new bacterial cell, it is known as generalized transduction. Transfer and packing of pure bacterial DNA. If we revise all these steps, there are total 5 steps in generalized transduction. In first, the bacteriophage is simply react on the bacterial cell. The bacteriophage transfer its genome into the bacterial cell. Ultimately, lactic cycle begins, and all the genome of bacterial cell as well as phage virus break down into small pieces. In the next step, the scattered chromosomal pieces of both of the species started to gather, and the virus genome, that is when the bacterial cell formed the new great number of viruses inside the bacterial cell and ultimately rupture the cell wall of bacteria and escape. Few of the new viruses captured the chromosome of bacterial DNA instead of their own. So when these muted bacteriophage affect and other bacteria, they ultimately transfer the genome of bacteria from one bacterial cell to another bacterial cell instead of the virus transfer. So what happens? The recombination occurs. The new recombinants forms. The new combination of DNA is happen. Let's just discuss another type of transduction, which is specialized transduction. What happens in specialized transduction? Suppose what happens if the bacteriophage genome, when transferred into the bacterial cell, it does not start the lattice cycle. It does not destroy the genome of bacterial cell or completely separate into the fragment, but instead of that, the genome or the DNA part of bacteriophage starts to integrated into the genome of bacterial cell. It becomes part of the bacterial cell genome. Then what happens next? Simply, the genome of bacteriophage that is integrated into the bacterial chromosome is start to separate out. Occasionally, prophage DNA exit incorrectly, taking the joining of bacterial DNA with it. When the integrated part of the bacteriophage DNA is start to separate, it starts to exit, it exits incorrectly, and when it completely separated from the genome of bacterial cell, it takes some of the part of the bacterial genome with it. This part I'm representing it with purple color, and ultimately this part has a name A positive. It just take A positive with it. After the separation, the bacteriophage virus starts to replicate itself and forms the new number of copies of bacteriophages in the bacterial cell. Ultimately, the bacterial cell wall breaks, and all the bacteriophage new that is formed inside the bacterial cell starts to release from that, and they ultimately affect other bacterial cells. So what happens? So ultimately, what happens when they form few of the bacteriophages capsule the virus DNA along with the bacteriophage DNA, as I have already represented it here. So there is a mutation. It will ultimately cause mutation when it transfer to second bacterial host cell. In the next step, In the next step, when this bacteriophage, this one, when this bacteriophage is transferred or is transferred from the affected bacterial cell and have the chance to affect another bacterial cell, then it affects and transferred all its genome to the new host bacterial cell, like this. And what will happen when the new bacterial cell has no genome, A negative and B negative, has both of genes are absent, A and B, in that bacteria? What happens when the bacteriophage transfer its genome along with the A positive part? Ultimately, this part of A positive will integrated into the genome of bacterial cell, and what will happen, it will convert, A minus will convert into A positive, and the new genome of bacterial cell will be A positive B minus, like this. Now the new genome of host bacterial cell will be A positive and B minus. So it, the bacteriophage causes the recombination or the mutation of genome of bacterial cell. So this is the specialized transduction. Basically, recombination is taking place here with the help of transduction. Conjugation and transduction, they all do the recombination, the processes of recombination, but only the process, the type of process is different. So we can define the specialized transduction as the transfer and packing of small percentage of bacterial cell, bacterial DNA with the viral genome. The viral genome is the red one. The packing of or the transfer of small, very small percentage, A positive, we represented it here as A positive, small percentage of bacterial DNA with the viral genome when transferred and packed is known as the specialized transduction. So this is very easy. Plus there are two types of cycles, there are two types of processes, phenomena in the bacteria. First one is the lytic cycle. Second one is the lysogenic cycle. The lytic cycle is the one in which the bacteriophage genome completely destroyed the bacterial DNA or bacterial genome into small fragments. This is the one I'm representing it here. Lysogenic cycle is the one in which the bacteriophage genome is become part or it is integrated into the bacterial DNA. This one. So here are two types of cycles, lytic cycle and lysogenic cycle. So I hope you understand the lecture completely and you have a very clear concept of lytic cycle, lysogenic cycle, plus what is generalized transduction and what is specialized transduction. So if you have any question, please do ask me in the comment section. Thank you. Allah Hafiz.